Hello, everyone, and welcome back to the molpigs podcast. Today, our guest is Josie Kishi, who will be telling us what excites her about molecular programming now and in the future. Also with me today is Anastasia.
Hello.
And I'm Will. Josie got her BS in computer science at Caltech and her Ph.D. at Harvard, where she was advised by Peng Yin. During her Ph.D., she developed the primary exchange reaction method, or PER, or PER, as well as being able to perform logic and record temporal molecular events. She has developed the related SABER method for multiplexed tissue imaging. Josie is now a technology development fellow at the Wyss Institute. Josie, hi.
Hey, thank you both for having me today.
Thanks for coming. For those who are new to PER, could you tell us a little bit about it and how it works?
Yeah, definitely. So PER stands for primary exchange reaction. So it's a very basic kind of mechanism for synthesizing DNA sequences. So you might have heard of toehold exchange reactions, where roughly speaking, you're able to kind of swap these short toehold domains, maybe 7 to 10 nucleotides for one another via strand displacement. In this case, the primary exchange reaction allows us to basically, given a specific sequence domain, let's say maybe 9 nucleotides on the 3' end of a strand, we can add another specific nucleotide domain to that. So let's say we have a sequence X, we want to add a sequence Y to it, only if it ends in X. That's kind of the logic we follow. So we can sort of cascade these reactions together to assemble these longer and longer sequences of DNA. And we achieve this by combining principles of strand displacement, toeholds, and using a polymerase enzyme.
And then how does that lead into SABER?
Yeah, so in general, PER is a very general mechanism for kind of dynamically assembling these strands, these sequences. So we've used them for a lot of different aspects. So we can make this synthesis conditional, for example, on the presence of specific signal strands. So in this way, we can program logic. We can also make it sort of program and record temporal interactions. So if different signal strands are present in different orders or different times, we can record that into the sequence. And then we can also use it as kind of a more general technique for synthesizing long repetitive sequences that we call concatemers. So we can repeatedly append the same sequence domain. For example, sequence X, it could end up starting with one copy of X, and you can end up with a whole long concatemer of X, 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 X. And what we use that for actually for imaging is for signal amplification. So there are a number of methods that allow you to visualize where DNA, RNA, and protein molecules are in biological samples. So either fixed cell samples or fixed tissue samples. And what we can do is attach these long repetitive sequences to nucleic acids that are localized, for example, to specific RNA molecules. And then using these repetitive sequences, we can use a complement, so X star, for example, that has a fluorophore on it that localizes to the X sequence. And because we have so many sites of X, we can localize many, many fluorescent strands to each individual concatemer. So in this way, we kind of localize and amplify the fluorescence signal in specific locations in the cell or tissue.
So you mentioned that with PR and like in your initial manuscript, you implemented some logic functions as well. Have you pursued that further from a theoretical perspective, or potentially for integrating with SABER and imaging for maybe more complex type multiplexing or calculations of different biomarker combinations or anything along those lines?
Yeah, definitely. So kind of all of the above. I think, I should mention Tom Schaus, my colleague, actually before PER developed a reaction called autocycling proximity recording. So in this method, he localizes these special hairpin molecules to, for example, two proteins that might be nearby each other. And using a related kind of synthesis reaction, he's able to develop these proximity records that say what specific individual single molecules are next to what other specific individual single molecules. So he uses that synthesis as a way to record molecular proximity. So that actually inspired the PER method, which also uses a very similar hairpin type construct. And so, in general, we actually have a lot of work in the lab using this dynamic synthesis for a lot of different types of recordings. So for the PER paper, we showed how we can sort of record what signals are present or even like what order they're introduced in. And this is actually something that can be extended even further So there's a project in the lab trying to use PRs, for example, a ticker tape that can actually record information that happens, sort of events and information that happens over time into these growing strands of DNA. And what's nice about this, right, is that you can easily sequence the DNA strands you produce. So you can convert those records into sort of a digital format through sequencing. And there's also other work that's using these types of cascades to record even more complex spatial configurations with the hope of ultimately, again, recording this information into sequenceable reads.
So for this kind of temporal recording, what sort of timescales are we looking at?
Yeah, that's a great question. So it's all kind of based on strands interacting with each other. So you can either imagine it as kind of a diffusive paradigm, in which case you might think about kind of the on rate of the molecule, which will depend on the concentration. So, that could be anywhere on the order of seconds to minutes or even longer, depending on what your concentration is. But there's also ways to do, the sort of surface localized experiments. So like the autocycling proximity recording I mentioned, that's happening with molecules that are right next to each other. So their effective local concentration is quite high. So that would probably, that could be pretty fast. Probably nothing like below a second for any of the kind of things we're looking at recording, but we're thinking of recording things that happen on longer timescales.
So like minutes to hours?
Yeah.
So presumably that would be really useful for recording concentration changes over time, say in cells or similar.
Yeah, concentration changes or configuration changes. I think this is very aspirational and I think we're quite far from it, but it would be awesome if you could, for example, attach a primer to like a ribosome, right? And then you could record like what RNAs are being, produced nearby or like what proteins are being translated, all of that kind of stuff. And you could try to understand, the temporal and the spatial dynamics via just sequencing of DNA records. Now, of course, we're very far from that at the moment, but I think that's kind of the aspiration.
Yeah, that's really cool. So yeah, like in that sense, in terms of more long-term versus short-term things, you've developed technology like SABER for biological imaging that is useful in labs today and has really immediate practical applications. How do you balance short-term goals with more long-term ones, maybe perhaps less immediately practical ones?
Yeah, I think that's super tricky, and I think that's also, this aspect that you have these sort of theoretical, aspirational goals, and then there's the sort of more immediate experimental goals is one of the interesting dynamics in the molecular programming community as a whole. I think a lot of it comes down to the PI, the lab, the institute, and kind of what the established goals are. So, for example, here at the Wyss, we're a very translationally focused institute. And Peng especially is very focused on these sort of practical applications. So we've been focusing a lot on using, these reactions, maybe the simplest form of them, to do really useful practical things like imaging or even adapting them for diagnostics. But I think that depends a lot, on the lab and on the researcher as well.
But for you personally, are you happy with the balance you've been having in terms of long-term versus short-term?
Yeah, for me, I've been very happy. I came from a computer science background. At Caltech, I took Eric Winfrey's molecular computing class, an elective sort of computer science course, and that got me really excited about the theoretical aspect. And then, I was kind of debating what I wanted to go into in grad school, and that seemed like a good time for me to kind of switch gears and learn something new. So, when I got to grad school, I sort of barely knew how to pipette, didn't really know that much more. So that was a great opportunity for me personally to learn, all of the basics of sort of molecular biology protocols and like how to think about these in practical terms. And, of course, like how to decide what to spend your money on, like what strands you want to order. You want to try 100 things, but maybe you can only, buy things for 10. So how do you decide like what's the most likely to work? So all of that kind of more experimentally rooted thinking is something that I've spent the last several years learning. So I've really enjoyed that myself.
So was Eric's elective course the first exposure to the field, or had you heard about molecular programming before then?
Yeah, it's interesting. So I think that was my first like concrete exposure. But of course, like I'd heard sort of from other students, like that the course was really cool and there were all kinds of like you could solve like the traveling salesman problem or whatever with DNA strands. I was like, that's crazy. So I'd heard about the class, I'd heard about some of the concepts, but I didn't really know what it was all about till I took the class.
And so in that class, presumably, and your background in Caltech, as you mentioned, was more on the computer science side, and like with PR specifically, like you've shown that you can construct logic using PR. Have you developed that further to maybe go back to where you started and like see if you can build up bigger circuits and more like a DNA computing side to PR beyond the like more biological applications?
Yeah, it's definitely something I've thought about, and I think other groups have also thought about kind of similar concepts. So one of the ways I like to kind of abstract away the reaction, right, is that if you think of the three prime domain, so the three prime end of your growing strand as the current state of that strand, and then the sequence you add represents like the next state. Then you can think of a PER reaction as being an edge between two nodes from a current state to the next state. So you can imagine like the sort of set of PER reaction cascades you can program can be sort of abstracted into these sort of nodes with different edges in between. So we're basically controlling, what states can lead to what other states. And we can make those edges, those state changes conditional, for example, on different signals that are present, um, or things that are happening in the solution. And so at a kind of more general level, you can think about both the sort of set of programs that you can write with this type of node network, but also like what types of programs you can't solve. And there's a lot of computer science theory around that. It's I think quite related to these finite automata that sort of are very simplistic, but they change state, and you go between different states. And there's a certain amount of information that you can process, and then some types of information that you can't sort of process or solve.
Yeah, I was going to say that sounded quite reminiscent of finite automata. So have you been able to show that PER kind of is as computationally powerful as finite automata? Is it more or less? What can you tell?
Yeah, it's interesting. I mean, I don't have any like sort of proofs, but that's definitely how I sort of conceptualize it. Um, in my head, right? And I think it's a nice way to think about it too, because it helps sort of motivate these recording applications, right? Because not only are we sort of having these, billions of whatever little automata going around changing state based on their local environments, we're also recording all of those state changes into the growing strand of DNA. So if it goes from state A to state B, then back to A, then to state C, right? That's all encoded in a specific, permutation of sequences that we can then recover with sequencing. So I think it's a fun way to think about what types of recording and what types of problems we can solve with those constructs.
How noisy are the recordings, and how feasible is it to actually extract like the fine-grained information that you've been describing?
Yeah, I think that depends entirely, of course, on the application. So I think the most challenging is the sort of temporal recording, and we've got a lot of collaborators and other people kind of leading that project, looking at the kind of math aspect of it. So how do you fit these times, right? If you think about at the molecular level, right, it's a stochastic random reaction, right? And so, it has a certain rate, but if you look at an individual molecule, the actual number of sort of incorporations you might have over a specific set period of time is random, but it follows a predictable distribution based on the rate. And so you can use basically fancy math to kind of back out, deconvolute those signals. But I think that's probably the most sort of challenging, especially because you're right, like there's also like all kinds of noise from other things that we don't think about. So like, we think about it as a very simple reaction of like, the hairpin and the primer come together and then they go to a state, right? But actually there's a polymerase involved, and like, what's the polymerase dynamics there? How fast is that binding? How often? Or even more simplistically, right? Like you have a polymerase that binds and the nucleotide has to be there and it has to be added. And this has to happen, 10 times to add, a single sequence domain. So that's actually like very complex to model. But then for other things, so like the autocycling proximity recording that Tom worked on, that's more, I guess, discrete. So, you get a record that says like this molecule was near this molecule. So you don't have to do quite as complicated of math.
On the topic of challenges, what do you remember any particularly difficult or challenging parts when you were coming up with the reaction, or did it kind of come fully formed into your head one night and you went from there?
Yeah, I think so. I actually got really lucky, with my colleagues. So I wasn't actually the one that invented PER. So Tom, like I said, he worked on this APR reaction ahead that really pioneered this idea of a hairpin that can pattern the addition of a sequence domain. And then I think it was actually my colleague Feng and also my PI Peng who realized, like, oh, if we apply this concept of a toehold, so this idea that the strand could bind and then come back off and fall off, then like we might be able to cascade these together into even more those sort of larger circuits. And at the time, I had just like officially joined Peng's group, but I was kind of exploring different project ideas. And actually, Peng came to me. He's like, "Hey, I'd like you to work on this," and I immediately, I immediately got it right. I was like, "Oh, this is super exciting. This is like, a new what we call molecular primitive, right? It's a very basic way to mutate information. We're exchanging one primer sequence for another, and like that could open up so many doors." So I was super excited to be able to work on that. And then, of course, the challenge came of like, where do we start? How do we do it, right? And so that was a lot of months of optimizing, like how long should these sequences be, uh, like what should they be? How should we implement the stopper behavior? So part of the design of our hairpin, we have to have a molecule, like a modification that stops a polymerase so that the primer copies some sequences and then stops. And then we have a competing strand that displaces it back off. So we have to think about, okay, what kinds of chemical modifications can we use to actually stop the polymerase? So the thing we kind of settled on for most of the applications initially is actually a three-letter code. So we include, for example, just the A, T, and C bases, and then we exclude G. And then if we have a C base on the template of a strand, the polymerase gets there and it has nothing to pair with the C, and then it stops. So that's actually turned out to be one of our most effective stoppers. But there were lots of tricky things involved too. So one of the things sort of learned about empirically is that the polymerase we use, BST large fragment, it has this A-tailing behavior. So when it reaches the end of a normally blunt end, it'll add a single A base overhang on the end. But it seems like this even happens in the context of our hairpins. So it adds an A base. So we found out that the efficiency, like the sort of yield per reaction, goes up a huge amount if you accommodate for that in the next step by always starting your next primer with an A, so that if it has A-tailed in the previous step, it can still bind and copy through the next hairpin sequence.
So, on balance, having done all this theoretical and experimental work, would you say that the more difficult part, well, which is the more difficult in your opinion, working out the experimental issues or doing the theoretical side of it?
I think they're just different, right? And I think they're different types of problems, different types of thinking, different types of approaches, right? I think, the from a like cost perspective, I guess the experimental problems can be harder to solve in the sense of like, if you're modeling something in silico on a computer, right, you could test like, I don't know, 1,000 or a million conditions, right? And then, when you're in the lab, usually that translates to like buying 1,000 different sequences or running 1,000 different reactions, right? Which either translates to cost of reagents or the cost and value of your time. So it's a different way to approach problem solving.
So did you find the transition, like going from mostly a theoretical background to, well, immediately presumably jumping into the experimental challenges as well? What was that something that you found fun and invigorating, or was it like more of an imposter syndrome type situation, or a bit of both? So presumably that's relevant to a lot of people in our field because of just how interdisciplinary it is. You always find yourself in a case where you probably feel like you don't know what you're doing.
Yeah, definitely. I think I'd say it's definitely a bit of both. So, and I think brings me back to why I really like the molecular programming community is that I've been very fortunate to have really great mentors, both on the sort of theoretical side and experimental side, that have really supported me, sort of despite my like complete lack of experience in the particular field, right? So after I took Eric's class, I realized that I was like, molecular programming, like this is what I want to do with my life, but I have like no experience, right? And I was very fortunate that Niles Pierce actually let me do some work in his lab. So that's where I learned to like pipette, um, and also like see how people approach the experimental problems. And then somehow I was able to get into grad school, and that was also very fortunate, right? And I think Peng's group, but also a lot of groups, we have people from lots of different backgrounds, so like computer science, physics, math, chemistry, bioengineering, and people have generally been very supportive of teaching and sharing their experiences and the methods. So I think, I've been very lucky in that sense to have great mentors. So I think that's something that I've always really liked about the molecular programming community, and I hope that can continue even though the molecular programming sort of project grant has ended.
Yeah, I fully agree that it's like the community is just super friendly, and personally, I feel like it's just a lot of fun, and most people are doing it because it's fun. Um,
Yeah.
but yeah, as you've been mentioning, like there are also a lot of really exciting practical applications, which can be huge drivers for things going forward.
Definitely.
In that sense, do you think that it's more of like, well, these are early-ish days for molecular programming and we're all having fun? What do you see going forward? Will the focus shift more towards like developing technologies that can be, more real-world applications, or do you think this more theoretical drive will stay on as well?
Yeah, I think I guess I keep saying this, I think both, but I think what I hope to see going forward is like that kind of community expand. Right? Like molecular programming isn't so much of like a specific set of methods or ideas, right? It's like really like an engineering approach and a way to think about biology and biological parts, and how you can hack them, how you can put them together to do these crazy new things, right? And it's also about community, right? A group of people who have come together with this common sort of vision of programming biology like computers, right? So I guess going forward, I hope that actually, the theoretical experimental work goes forward. I hope that we can also find ways to kind of bridge that gap and just kind of expand into new areas. I think one of the most exciting areas for me personally is seeing the DNA data storage stuff really take off. We have all these different ways to mutate, rewrite, exchange information, so total exchange reactions, like tile assembly, like primary exchange reactions, like other enzymatic and non-enzymatic cascades. And then we already know that nucleic acids can store this insane amount of information, right? And so like seeing how we can put those ideas together to really have not just write lots of information, but rewrite it and access, retrieve that information, I think is really cool.
Yeah, did you see the announcement by, I think it was Western Digital, Twist, IDT, and Microsoft, where they're kind of putting all this industrial money into developing that? What are your thoughts on that?
Yeah, I mean, I think that's awesome, right? Like, it takes money to make the sort of experimental, practical aspects work. So, I mean, I think it's great to see those collaborations.
Yeah, and speaking of like fun things in the field as well, like it probably wouldn't escape anybody's attention. Like the really fun acronyms and names your lab tends to come up with for technologies you develop. Do you do that when you first start on a project, or after like, hey, this is going somewhere, we need to brainstorm a fun name, or like is it just something organic?
Well, surely what you do is first come up with the name and then you design the theory to fit it, right?
I think it's a bit of all of them, right? So like PER, I think Peng came up with that pretty early on, and I think it really stuck because it really evokes this idea that you're exchanging information by swapping out primers. So you could think of, toehold mediated strand displacement cascades. They use toehold exchange where they're swapping out these sort of different toeholds and different steps of the reaction, or like I often think of like the four-way branch migration as like a strand exchange reaction, right? So this sort of fundamental idea that you're exchanging information in a single reaction and that you can sort of generalize and cascade together, like I think that was really resonated with us. So that pretty much stuck from the beginning. And then SABER was a little bit different. So we had different, like we were kind of calling it like PR amplification or, amplifying the PER or whatever, right? And I kind of had it in my head that I wanted to call it SABER because, what we're doing is we're amplifying, like fluorescent, like light signal, right? And so it's like light signal amplification by exchange reaction, like by primary exchange reaction. And then I was like, oh, well, obviously this should be lightsaber, right? Which we later shortened to SABER. So it was just, you got to get the right timing of like getting all of your colleagues on board. But people were generally pretty excited when I brought it up. So it really pretty much stuck from there on.
Yeah, I guess the concatemers are kind of like rod shaped as well in some senses, so like just like wobbling around.
Yeah, exactly. Like, and it's like you're really like if you think about like lightsabers, right? You start with like the handle or whatever, and then it C2 grows the like fluorescent signal. So it's like perfect, right? Because I think it evokes like the kind of molecular mechanism almost.
It is.
Did you have to switch to the name Saber because Disney threatened to sue you for using lightsaber?
Well, I don't think they've caught wind of it yet, so but
Yeah, so it's okay if you're biased, but is lightsaber your favorite acronym in the field, or do you have some other favorites?
that's a good question. Yeah, I'd say I'm pretty biased on that front, but I feel like I'd have to look at like a comprehensive list because I think there's been some really great ones.
Are there any other projects in the field that you're keeping an eye on and trying to see where that might go?
Yeah, so one of the things that I've seen, like kind of another emerging trend, right, is using our ability, especially with like total exchange reactions, to specifically and very sensitively detect strands, being applied more towards things like diagnostics. So I've seen like Andy Ellington's lab and others have really started to do a lot of very cool diagnostic work while applying these kind of arguably molecular programming type concepts. So I think that's also very exciting to see.
Yeah, I guess like there has been an explosion in like diagnostics technologies these days in general. Do you feel like that will affect things in the long term, or like what will it bring people together working on diagnostics platforms, or maybe increase competition in some senses?
Yeah, I mean, I think I've seen a bit of both. So we've been doing some diagnostics work in the lab as well. And I think there's so much innovation in molecular programming and concepts of like what is like the theoretical limit of like how specifically we could detect a strand, like one strand from another. And, it comes down to like the thermodynamics of it. Um, and then, really applying that in the practical sense of diagnostics, I think is very exciting. Of course, like I think diagnostics is fairly competitive because you're sort of competing on like a few main fronts, so like cost, specificity, sensitivity, right? So I think you'll inevitably have some competition, but I think there's still a ton of space for innovation there.
You've got all these different applications and subfields. You've got diagnostics, assembly, storage, computation, so many different things. And I think we're really lucky that kind of we're still at the stage where we can all meet together in one place, well, virtually in one place now, and kind of get to hear all of these different sides and have it as a single session conference like at DNA. Do you think as our field hopefully grows and expands that we'll have to splinter off into different fields and we'll lose the cohesion, or do you think there's a way we can keep everyone together as it gets more successful?
Yeah, I mean, I think I hope it'll expand. I hope we can keep the DNA conference and F Nano because I think they are great opportunities to bring people together who have this vision of programming molecules, right? And that's something, of course, I've always loved is that the theoretical people are always willing to talk about their work and try to explain to experimental and vice versa, right? And there's really this openness of communication and mostly a lack of competition, because the field is, so new and there's so much space for innovation. It's not like people are like competing in like other fields, right? You might be like competing to get like the highest resolution or the most number of targets or something specific like that. But molecular programming, right? It's like you have so much rich innovation that happens in so many directions to go. You're not really competing. but yeah, I absolutely hope those conferences could continue, and also that we could sort of expand to other fields, right? I think there's a lot of overlap, of course, with like synthetic biology, and so like maybe communicating more and moving more in that field, right? And like sort of adapting these concepts of computer science, which we already see a ton of, right, in synthetic biology, and really opening up those communities and communication between those, I think, would be very valuable.
What do you think might be the most exciting bridge between DNA computing and synthetic biology? Where do you think the killer application for that is?
Yeah, that's a great question. I'm like trying to think of, I mean, there's all kinds of things you could imagine, right? I guess I think of like synthetic bio as like, specifically programming systems in like living cells, living systems, or like some kind of life-like system. So of course, like from that front, like being able to like create life or whatever. Maybe that's too aspirational, but like create a system that actually like, maybe like thinks like a brain or something, right? That like employs these neural networks, or like that can, interface with biology, or of course, like if there were any applications in like human health, like can you make these like smart therapeutics? Like, you see a lot of work. People are like engineering microbes that could like go in your gut and like fight off like bad pathogens or something, right? So like really expanding that to like, oh, well, what if you could like just record everything and then just fix everything on the spot? Like all that kind of stuff I think is really awesome.
Do you have sort of a more longer term vision in that sense, in terms of where you'd like to get involved? Because you said you want like continue working on molecular programming.
Yeah, I mean, I've so far managed to stay away from the in vivo stuff, which is, there's sort of like, in vitro, or like, I guess maybe you even start with like in silico, something like will always work, and then you go in vitro, and then like there's all kinds of weird bugs and like debugging that has to be done to like get it to work, and then like it's a whole nother ball game to go in situ, so like the imaging stuff in like cells and tissues, and then like I don't know, in vivo is like just way out, like way far from there. So I think for the near term, I personally am not planning to go in the in vivo stuff. But I think I've really enjoyed learning about these kind of in situ methods of like how do we image and record information about molecules in biology, and like can we use that to study biology, and then ultimately reprogram it based on sort of what we learn about what biomarkers are important for disease, or whatever, right? So personally, I think I probably will stay pretty squarely in that in situ space. Try to develop tools, based on different molecular programming aspects that can be generalized for studying biology, with the ultimate goal of like improving it.
And I guess when you move to in vivo, you need to contend with not only in silico bugs, but real bugs as well that are interfering.
Yep.
What's the craziest idea you've ever had with molecular programming?
Yeah, I guess I probably like these molecular recording applications. Are like pretty out there. I think they're maybe not the most aspirational, but they're the most sort of like concrete, sort of future goals, right? Like we have sort of the basics of the like physics and stuff working, that we think we could get there, but we're definitely not there by any means yet.
Yeah, you've had like a really interesting and successful journey in molecular programming so far. Do you have any advice for emerging early career people in the field? Anything you would have done differently, or anything to suggest?
Yeah, I think in terms of advice, let's see, like try to read a bunch of papers, put yourself out there, even if you have no idea what the heck you're doing, and don't think you can do it, you absolutely can do it, and like try to find a good mentor. I think that was really key for me, right? Having these mentors at Caltech that really supported my vision, sort of despite like a complete lack of experience. Um, I'm also really excited. I've heard some of the folks at Caltech are going to make an art of molecular programming textbook, and I think that would be really awesome as a kind of primer, I guess, to the field, and hopefully that'll make the content more accessible. Because I think there are still a lot of concepts that are quite esoteric, and it can be hard to really kind of get into the community. So I think um, having that textbook will be great, and then yeah, I think the big thing is try to find good mentors, maybe try to do, if you're a college student or even a high school student, try to do research over the summer, um, and just try to like talk to people and meet them, talk about ideas.
Yeah, we're all really excited about the art of molecular programming because you basically have to be an engineer and a physicist and a computer scientist and a biochemist and all of these things. Um, which would you say was the most challenging for you to get into, looking back?
Yeah, I guess for me it was definitely like experiments, like the experimental side, because I just had basically no background in that. I pretty much right up until junior year when I took the elective course from Eric, like I'd been planning to go into software engineering. So I think doing that transition was challenging, but on the other hand, I'm not doing in vivo work, so I guess it's not as hard as it could have been.
Do you regret not going into software engineering and getting the Silicon Valley salaries that would come with it?
Oh, not at all. Yeah. I think like, it's interesting because I applied for software engineering jobs at the same time as grad school. And so I really like sat there and thought about it. But I think, molecular programming is like, it's just so exciting. And it's like a totally different way to sort of think about computing. And, I was just excited to just have this opportunity to work in it.
Were there any big culture shocks when you went from West Coast to East Coast? Have you noticed any big differences between how people work?
Yeah, I mean, I think there's, kind of everything you might expect. Like um, I feel like people are a little bit more like serious here. I don't maybe that's just my like institute because we're very translationally focused. Um, but I think there's like we definitely focus a lot more on like, okay, what is the like practical application of this technology and how will we get there? What are the milestones and like how much will it cost? And I mean that's also very like lab dependent, institute dependent. Yeah.
And how did you find the difference between undergrad, postgrad, and now postdoc?
Yeah, let's see. So undergrad, um, yeah, again, I was very focused on like CS. So it wasn't until like kind of the very end that I was like, oh, actually, I'm going to go to grad school, right? But I think uh, so. In grad school, I'm in Peng Yin's department, so I was in systems biology. So that was actually very interesting because systems biology, kind of like molecular programming, right, brings together a lot of people from different fields. In the case of systems biology, you're trying to think about biology, from a sort of stepped back position where you want to think about it as a whole and how all the different parts sort of interact with each other, which is actually like very similar to molecular programming, except you're in most cases you're studying it, not reprogramming it. Um, so I think that was really awesome for me to sort of be exposed to this other community too, and see how like people like that think as well. Um, and then it depends a lot on your grad program of like how many classes you have to take and stuff. But I was actually lucky that we only had to take, I think, 4 classes for kind of like the whole time, and then TA one class. And so that left a lot of open time for research, which I think was really really valuable because then basically I could just hit the ground running. Once I joined the lab, and yeah, in my case, I'm still here after grad school, stayed on as a postdoc, and now a tech dev fellow. So obviously, I've really liked it. And my day to day has been, I guess, largely the same. I mean, lots of experiments. I've definitely taken a bit more of a sort of business development kind of spin on things because I'm really interested in trying to spin out some work. So I think, uh, but in terms of the day to day, I come to lab, I do experiments, I think about problems, and I try to execute and debug mostly. So.
Do you still mostly have control over your own time, or are you starting to have to think about spending 20% more of your time on grant writing and admin? How's the balance been for you?
Yeah, I think it kind of depends on the time of year and stuff. So I was lucky to have a chance to work on a bunch of grants in the past, so that was pretty cool to kind of think about how do you write up these ideas in a way that like convinces people that it's important enough to spend usually like taxpayer money on, right? And then more recently, since I'm really focused on spinning out some work, I've kind of switched to focusing on things like, working on our IP portfolio, and trying to like reach out to like investors and stuff. So, I think uh, it's always challenging to balance your time between the kind of, the BD, the grant writing, or like thesis writing, and then doing the actual experiments. So I think that's always kind of like a daily challenge. So I try to approach each day and think about, okay, what can I actually accomplish like today? But like kind of go from there.
Did working on the BD side of things, did that come to you as a surprise, or were you kind of expecting that from early on?
Yeah, I think so the Wyss Institute has a dedicated business development team. So we've got like BD directors and leads, and also like lawyers that help us with filing patents and things. So that's always been kind of a part of the work we've been doing in the lab. Um, so that's been really great to have that as a resource. And part of that comes with this idea that we're a translational institute that really focuses on getting these texts out there. And then, similarly, they've been extremely helpful in helping us like prepare pitch decks and like, uh, you know, reach out to people in different networks to talk to them about our ideas. So I think that's been an incredibly valuable resource to have.
What do you think the most imminent commercial application of PER or SABER might be, or is that still all under wraps?
I think it's still a little bit under wraps, but I think, with imaging, right, there's always this need to study not just what molecules are in a sample, but where they are, right? Because this can tell you a lot about what's going on. So you could imagine like having, if you have a tumor sample, you could look at how gene expression changes across, right? And try to understand like how it developed in the first place and like, what's going on at the molecular level. So I think, and what makes the amplification really helpful is that if you sort of increase the signal by like 100-fold, right, you can reduce the exposure time, so the amount of time it takes to image a region, by a similar amount, so like 100-fold, right? So if you can image 100 times faster, then you can image like a much larger tissue area in the same amount of time. And so I think those applications where you're really trying to again, sort of create these like large atlases, molecular atlases of tissues, I think are very important.
Do you think there are any big bottlenecks in the field that are kind of making it difficult to progress, and if so, what do you think might be the best ways to solve them?
One of the things I'd like to see, right, is sort of the community grow and become more accessible. So I think um, like one of the things that I've found very helpful sort of when learning about like tile assembly and stuff is like there's software packages that like help you visualize what's going on. So a lot of this sort of communications work where you try to really like explain or visualize, or animate a process, I think, are extremely helpful in kind of getting those concepts out there. Um, so I think like one of the things that I've been trying to think about is like, okay, how can I like make like an animation or a video that shows like what we're trying to do, even though it's actually, it might be kind of abstract, but there must be a way to like show it in a way that, anyone could understand it. So like thinking really hard about how we communicate the science, I think is important.
Yeah, I remember the animation. I think they were DNA 23 and DNA 25. Did you make those animations?
Oh yeah, so I made some of them, and then we also actually have a dedicated animator at the Wyss who made like the really fancy ones with like the dark backgrounds and the like wiggly primers and stuff. So she put all those together, which was awesome because having that as a tool to explain what's going on. They say like a picture's worth 1,000 words, but I think an animation is worth even more than that, right?
Depends on the frame rate.
Yeah.
Thank you so much for joining us, Josie. We had a great time talking to you and hearing all of your thoughts about the field and where you think it's going to go. And I hope all of these come true. Stay tuned for our talk with Yuan Chen in a couple of weeks' time. Thanks for listening.